Welcome everyone to the second part of our lesson. Okay, so our second part of the lesson has to do with blood group typing in forensics. Now, blood group typing in forensics is actually a very important part about the investigations, especially when we are dealing with blood samples that we have collected from the crime scene. Okay, so this is going to be a video on the different blood types, right, and how they came about in human beings, but I will not play the video. Okay, you can play the video on your own. I've included it inside as one of the links in the classwork material for this lesson. Okay, now first we are going to talk about antigens and antibodies. So from the video you have known, right, that red blood cells have proteins called antigens on their surface, right? Okay, so what are these antigens? Okay, they are basically proteins that serve as recognition markers. Okay, they serve as recognition markers. Okay, and they are protein in nature. Right, now these antigens will then be grouped accordingly lah, whether to their A, B, or O. Right, now plasma on the other hand, right, you should have learned this already. Plasma, the blood plasma, has antibodies. Now in case you have forgotten, right, our blood here, as you can see at the test tube on your left, right, can be separated into three parts actually, right? One down here is your red blood cells, as you can see, the red part here, and finally the beige clear part, liquid of your blood is called your blood plasma. Now in your blood plasma it contains antibodies which recognize and bind to specific antigens on the red blood cells. So antibody A is going to bind to antigen A, right, and antibody B is going to bind to antibody antibody B. Now take note how I wrote the antibody and the antigen. This shouldn't be antibody, it should be antigen, right? Okay, now take note how I wrote the letters of the antibody and the antigen. Antibody is always written as small letters and antigens are written as capital letters. Same here. Okay, let's cancel this. Right, so this is the basic thing you need to know for our blood. Our blood has antibodies A or B. Okay, sometimes both, sometimes don't have at all, depending on what blood type you are. Inside our plasma, our red blood cells, which are a separate layer, right, inside your blood, okay, they have the antigens there. So the antibodies are found in the plasma, antigens are found on the red blood cells. Now, how then do we distinguish between the A, B, AB or O blood groups? Right, so we have your A, B, and O right here. Now, for the A blood group, means your red blood cell has antigen A on it. So you can see all this antigen A on the surface of your red blood cell. And in the blood plasma of blood type A, you have antibody B. Now, why do we have antibody B instead of antibody A? Now, please remember that antibodies are meant to recognize foreign cells, right? So if you have antibody A here, then you recognize antigen A. That doesn't make sense. Then at the end of the day, your immune system is going to attack your own red blood cells because they now recognize this, right, as foreign, right, because these are meant to identify things which are foreign. So that is why for blood group A we have antibodies B instead. Why? Because we want to kill anything that is not A, right? Anything that is not A, okay, we want to destroy them because they are seen as something that is foreign to the body, right? So let's take a look at at blood group B instead. So for blood group B, we do see antigens B on the blood surface, on the red blood cell surface, and then we have antibody A. So now you can see there's a trend here, right? So long as you are that specific, for you to be classified as a specific blood group, A or B, it depends on the antigen, right? And whatever blood group you are, you will have the opposite for the antibody. So if I have antigen B, I'm going to have antibody A. And for blood group A, I have antigen A, I'm going to have antibody B. Right? So this should be straightforward enough. Then finally we have blood group AB, where there is both antigens A and B on the red blood cell surface. However, they have no antibodies. Now why they have no antibodies is because if I have antibody A or antibody B inside my plasma of the blood group AB, then I would be attacking myself because I have A and B on my surface. So your body has evolved, if you are blood group AB, to not have any antibodies inside the blood plasma. Okay. Then for blood type O, instead of having any antigens, they have no protein antigens, right, of the AB type at all on the surface, and they will have both antibodies A and B, right? They have both. Now, if they have both antibodies A and B, this makes sense because anything that is foreign to O, let's say I put blood group A, right? This blood group A, I pass my blood to O. So what is going to happen is my antibody A and B, or should I say my antibody A, is going to recognize and stop this. Okay, so I will recognize the blood group A, and I will destroy the cell. So this is a protective mechanism to prevent foreign blood from invading into the system. Okay, so now what happens when different blood groups are mixed? So we have touched a bit on that previously. So let's say we have type B blood, which means we have antigens B on the surface, and that would also mean that we have antibody A in the plasma. Remember that they are opposite, especially for the specific blood type. So what happens when type A instead is added into your type B, right? And remember that type A blood has A antigens on the red blood cell surface, and we have antibodies B in the plasma, right? So what is going to happen is that the antibody A in the plasma, right, from type B blood, okay, the antibodies A are now going to bind towards your A, like so. So you can see the animation right there, right? So when they bind, what is going to happen is that agglutination or clumping is going to happen. Now when this occurs, right, you are going to see your blood being clumped up, okay, into thick pieces like this. Now you may remember that your blood, or your normal red blood cells, if they are in their normal liquid form, is going to be like a normal liquid, a normal thick liquid. However, when they clump, you are going to start to see clumping like this, okay, and you can actually see this under the microscope. Okay, now how do they happen? Is because the antibodies can actually link up one red blood cell to the other if they can recognize two separate proteins from separate red blood cells, right? Because they have two arms that you can see here, right? So one can bind to one red blood cell, another one can bind to another red blood cell, right? So during a blood transfusion, the effect of the recipient's plasma on the donor's red blood cells is considered. Now this part I'm not going to go through too much, okay, because this is more towards blood donation, but it's just for your own knowledge, right? So there is a reason why when we are blood group A, right, we can when we are blood group A, we can receive from A, we can receive from O, and we cannot receive from B and AB because we know that B has antibody A, right? Sorry, wrong. Is because my B here has antibody A is sorry, my antibody B here is going to attack this. Okay, same for blood group B. We have antibody A. That is why I cannot receive from A because I am going to attack my antigen A on my donor. Right? I can accept from B. I cannot accept from AB. Why? Again, because I have an A here. So I'm going to attack here. Right? Blood group AB can receive from everybody. Now, why this is so? Because they have no antibodies. Right? So that is why they are called universal recipients. Okay? Means if you are blood group O, consider yourself lucky because you can receive blood from all types. Okay? And finally O. O is what we call a universal donor. Why? Because you see O, I can donate to O. And if I go back, okay, so if I go back, I can see that they can donate to AB as well because I have no antigens. I can donate to B and I can donate to A because I have no antigens. Okay. Now, you might be thinking, Mr. Hakim, why we've been talking about the blood group typing for quite some time. So how is it relevant? Now, the principle of agglutination, right? When we, the blood groups actually clump up, okay? They are going to determine what is the blood type of the blood sample. So, when we go and take an unknown sample and we add a specific antibody towards it, we can then tell what blood type it is depending on whether it clumps up, right? And this experiment, right, these two experiments, we will be doing it, right, once you come back after your end of year examinations. Okay, so we will talk more about the experimental side, the experimental principles behind blood group typing in forensics when you have a chance to do the experiment itself, right? But for now, just know the concept behind antigens, antibodies that are found inside our blood. Okay, so modern crime labs nowadays more or less use DNA tests, but blood typing is still used occasionally, right? Especially when DNA tests are not available, because we know that DNA tests are the most accurate. Okay. Right, they are the most accurate form of identification because you can tell the exact sequence of the DNA that is unique to all individuals compared to blood types. Blood types, I mean, not everybody will have different blood types, right? You can have the same blood type as your friend, for example, right? Okay, so that's all for now. Okay, any clarifications you can ask them, right? To seek your clarifications. At 4:15 p.m. during the Zoom session. So do not be late. And after that clarification session, we are going to have a question that I'm going to pose to all of you, and you're going to try to solve it, as well as a Kahoot quiz at the end to wrap up the entire lesson. Okay, thank you.